Good afternoon, everyone. Today I will talk about enzymes, certainly about different types of the enzyme, additionally, factors affecting their function. Let's start now imagining certain types of the enzyme and how they will bind to our receptor and what the mechanism they work through it in our body. Another theory appeared in the past which explained the mechanism of enzyme working is called lock and key theory. In this theory, we are talking about enzyme and substrate like key and lock. The enzyme contains a certain active site for which the substrate is bind, like the lock and key. After that, there is update on this theory, which is called substrate or induced fit model, in which the enzyme active site will change its shape based on the interactive substrate. Once the substrate is fully locked in the exact position, the interaction or the catalysis can begin. Let's talk now about types of enzyme. There are many different types of enzyme based on their function. First one is called transferase, oxidoreductase, isomerase, which make an isomer, hydrolase, which make hydrolysis process, lysase, and ligase. The first type of the enzyme is called transferase. From its name, it's called transferase, which means that it transfers a certain molecule or a certain functional group between two molecular agents, such as alanine or phosphatic group. For example, we have an enzyme called alanine aminotransferase, which transfer or shuffle amino group between two amino acids, which is alanine and aspartate. There are another type of transferase enzyme which transfer phosphate group between ATP molecule or other compound. For example, in the formation of disaccharide, such as hexokinase enzyme in the glycolysis process. Second type of this enzyme is called oxidoreductase, and as it is illustrated from its name, it performs oxidation and reaction based on the substrate it works on. This enzyme performs oxidation and reduction, and hence it's called oxidoreductase. When the substrate is being oxidized, this enzyme acts as a hydrogen donor. On the other hand, when the oxygen atom is in the acceptor, these enzymes are called oxidase. The third type of the enzyme is called isomerase. Isomerase is an enzyme which converts the substrate into its isomer. But let us ask ourselves what it is isomers. Actually, isomers are molecules that have the same molecular formula but have a different arrangement of the atoms in the space. For example, when we have a molecule, it is formula called AB, the isomer will be BA. There is only one substrate yielding one product. This product has the same molecular formula as the substrate, but different in bond connectivity or spatial arrangement. Isomerase catalyze reaction across many biological processes, such as in the glycolysis and in the carbohydrate metabolism. Let us talk about another type of the enzyme which is called lysase. And from its name, lys mean to break something. So lysase it is a type of enzyme which is responsible for analysis for certain compound, such as decarboxylase. It lyse or break or remove the carbon dioxide group from the pyruvate. So in another word, this enzyme catalyzes reaction where functional groups are added to break double bond in the molecule, or where double bonds are formed by the removal of the functional group. Other example of this enzyme, deaminase, which remove the amino group, or rehydratase. When we hear the word ligase, we will think about ligating the things to each other, which mean binding the things to each other. On the other hand, the ligases enzyme do the same thing. It binds the things to each others, ligate it to each others, so it will work by the opposite mechanism for lysase enzyme. For example, where the hydrolase break bond by adding a molecule of the water, ligase remove this molecule of water to ligate the compound to each other. There are different subclasses of ligase, which involve the synthesis of the ATP. Now we will talk about the hydrolase. This enzyme catalyzes reaction that involve the process of hydrolysis. They break single bond by adding water. Some hydrolase enzyme function is as a digestive enzyme to break the compound by adding a water molecule. Hydrolase can also be a type of transferase, as it is transfer a molecule of water to another compound. For example, glucose 6-phosphatase, which remove phosphate group from glucose 6-phosphate and leaving the glucose with H3PO4. So now we finished talking about types of enzyme and how they interact with substrate either by the key and fit model and induced fit model. Let's start now to talking about factors affecting enzyme function, like concentration of the substrate, pH, temperature, and inhibitors. To begin with, saturation of the substrate, we will think that when we add more substrate, the speed of the reaction increases. But actually, this will happen with enzyme to certain up point, which mean that enzyme have a saturation point. Above this point, the enzyme velocity will be the same because it is fully saturated. When the reaction began, the velocity of the enzyme action keeps on increasing on further addition of substrate. But when we reach the saturation point, where substrate molecules are more in number than the free enzyme, the velocity remains the same. Another substrate-related factor which affect the enzyme activity is the type of substrate. There are different types of substrate, classified into two categories. The normal substrate which bind to enzyme for its active site and exert a certain action, and the other type is called competitive inhibitors. Inhibitors is a chemical which compete with the specific substrate on the active site of the enzyme. They are structurally resemble the specific substrate of the enzyme, but they have antagonistic activity, which means they antagonize the action of the enzyme. This concept can be used for treating a bacterial infectious disease. Each pH has a favorite pH at which the enzyme will work as a full capacity. Every enzyme will have its own optimum pH. Some enzymes favor acidic pH like pepsin or digestive enzyme, which work at pH equal 2. Another enzyme will work at the function or pH equal 7 or more, such as amylase enzyme in the saliva function. Any change in the pH will weaken the interaction that hold the protein to each other, which cause denaturation for enzyme. This means the enzyme will not function again. As we all know, raising the temperature will increase the speed of the work. However, this is well accurately with the enzyme, because each enzyme has an optimum temperature, above which there is destruction in the structure of the enzyme, in which the enzyme will be denatured and hence will not work. Raising the temperature generally speed up the reaction, and lowering the temperature slow down the reaction. However, extreme high temperature can cause enzyme to lose its shape. There is a certain temperature at which most of the enzyme are work, which is the normal body temperature, which is equal 37 degrees Celsius. And finally, we have to talk about the inhibitors. Presence of the inhibitors will affect the enzyme activity because it is bind to the active site of the enzyme, and subsequently the substrate cannot bind to this active site. In conclusion, the enzyme cannot be work because the substrate will not bind to its active site. To sum up, we talked today about the enzymes, certain types of the enzyme like ligase, hydrolase, lysase, and isomerase, and we talk about factors affecting their function like pH and type of the substrate, temperatures, and the presence of inhibitor. In conclusion, this is the name of the group member. You can find our reference in the next slide. Thank you so much for your listening.